of the year, actually. David Sweet and I are really pleased to see how many people came out for science today. Usually we do a lot of author stuff and other things, but I think it's our first real serious science lecture. So thanks for coming. Before I introduce Doug, I just thought maybe I should run down what his company does, what AbbVie does for the biopharmaceutical industry. So they are the leaders in 4 or 5 different areas. They have immunology, which is what they do for developing treatments for autoimmune diseases, such as Humira, which is for rheumatoid arthritis. They have neuroscience, which they focus on therapies for neurological disorders like Parkinson's and Alzheimer's. They also have virology, which is treating things like hepatitis C. They have some eye care products for glaucoma, and then they do obviously oncology. They have blood cancers and solid tumors is what Doug works on. They work on non-Hodgkin's and multiple myelomas. In the solid tumors, they use antibodies for lung, ovarian, and gastroesophageal tumors. So today, Doug is going to talk about his work in those areas. He graduated from the University of Pennsylvania and finished his graduate work at Northwestern. He works on the oncology team, but prior to that, he worked on type 2 diabetes. So currently, he's working on novel therapeutic antibody therapies and combination antibody and toxic drug therapies, which we'll hear about. So welcome, Doug. Thank you for coming.
Okay. Wow. This is nice to be in front of all you. Usually I see you on a paddle court or at a tennis court. So it's a real pleasure to be here today to talk about my work in oncology. When Jed and I have been talking about this for maybe a couple of years, he said it'd be really neat to have you come and talk about what you do in cancer research. And originally I thought, well, maybe I'll talk about different vistas and the new up and coming areas of cancer research therapies that could be exciting. And I started digging in on it. And I'm like, there's no way I could do that justice in 40 minutes. So I decided to change paths and really just focus on a specific new therapeutic area that I actually work on and have experience in. And that is antibody drug conjugates. And so my title of the talk is that it's a promising therapeutic strategy in the war on cancer. And let's start. So before I get into, so I thought just a quick review of what is cancer. I think everybody in this room has either been directly affected by family members or people that have cancer or relatives and just know the horrible burden and profound suffering that this disease causes. It's been termed the Emperor of Maladies. There was a book that came out. But basically, what I just want to review here is that it's a group of diseases that is marked by dysregulated or uncontrolled cell division. It's the second most fatal disease in the world, approximately 8.2 million deaths worldwide per year. Cancer is primarily caused by something called mutations, and these are changes or errors in the genetic code in cells and typically these are mutations in genes that affect cell division and cell cycle regulation. And these mutations can be inherited from your parents, your grandparents. They can be randomly generated. They can, but typically often caused by different environmental stresses. So things like sunlight, which can lead to melanoma. It's UVB that typically causes that. Smoking, obviously connected to lung cancer, air pollution, alcohol, radiation exposure, or other chemicals that are carcinogens. I pulled this diagram from a paper my boss thought was really good, and it really nicely summarizes the different multiple areas that are the primary drivers of tumor growth. And so I'm just going to kind of walk you through this, starting at the top. So some of the issue, and all of these involve mutations in these different kind of components that lead to these things that promote cancer growth. So in the top is proliferative signaling. This is there are things called receptors in the membranes of tumor cells, and when you have mutations in those things, it can cause a constant signal to the cell to divide and divide and divide, and that's where that condition comes from. There are also now going to the clockwise. The next thing is evading growth suppressors. So there are genes that actually suppress tumor growth, and if you have mutations in those genes, it's like you've taken the brakes off, and so that can promote tumor growth. Moving down, you have mutations or problems in active that cause activating invasion and metastasis. So these are the genes that enable a primary tumor to have cells from that primary tumor to then migrate from the tumor and then form metastatic lesions elsewhere. And that's typically how people die from solid tumors, is the dissemination of metastatic lesions. Moving down, you have on the bottom there problems or genetic defects in things that cause what it's termed loosely replicative immortality. So cells typically have a lifetime and they can divide a certain number of times. And there are certain gene sequences that get that are aberrant in tumor cells where this just never stops. So it's like it just constantly go through replication. And then now going up toward the red box, you have, this is more of a mechanistic feature, factors that induce angiogenesis. Angiogenesis is the formation of new blood vessels. And obviously, as a tumor grows, it needs to get more sugar and nutrients and oxygen so it can sustain its growth, because cell division is a very energy intensive process. And so that's another area that can, when vessels grow, that can promote tumor growth. And then the last thing in the gray box in the upper left there is mechanisms that resist cell death. So there's a interesting phenomenon called apoptosis, and that's a basically is programmed cell death. And tumors have mutations in genes that regulate that, so that it never has programmed cell death. So it evades programmed cell death, and that also promotes cancer growth. So I know that's kind of a lot to dump on you. Does anybody have any questions on that? That makes sense. I'm sorry. I'm used to talk. I talk to people because when you're doing this every day, it seems like second nature. So I know I'll slow down if I need to, and if there's something that needs to be further explained, I'll try and do it as best I can. But all right, moving on. So just more information on how complex and heterogeneous cancer is. There are a lot of different types of cancers, as many of you know. These can be broadly characterized as solid tumors or hematological cancers. So among these different types, you have things like carcinomas, melanoma, sarcomas, and these are really defined by the tissue of origin that they come from. And then you have like your hematological cancers, the lymphomas, leukemias, and then you have nervous system tumors, which are like GBM, which is glioblastoma, which are cancers from the brain and core tumors. And this is just a brief overview of that. I'm not trying to go into depth on this because that's really not what I want to talk about too much, but I want to just establish a little background here. And if anybody wants to chime in on that, anyone who's got a background in cancer, please feel free to do so. So, moving on, traditional cancer therapies, typically up until about 40 or 50 years ago, were primarily treated by surgery, radiation, or chemotherapy. Among these therapies, chemotherapy really has been the primary interventional strategy to treat cancer. And so chemo agents are basically cytotoxic agents that preferentially kill rapidly dividing tumor cells. The mechanism of action, typically with most chemo agents, is that it works by inhibiting DNA synthesis. And so some of these things like 5-fluorouracil, thioguanine, I know it's a mouthful. These are just examples of things that inhibit DNA synthesis, or they are DNA interacting or alkylating agents. So things like cisplatin, which is used for breast cancer, actinomycin D, anthracyclines, zinc alkaloids, modify the DNA in a certain way so it prevents that DNA from being replicated. So it basically stops cell division that way. Okay, so now chemo agents have evolved and demonstrated improved treatment outcomes over the years. However, they're still, as a strategy, it's hampered by several factors. And these include limited selectivity against cancer cells. So the chemotherapy doesn't just go to the cancer cell, it'll go to other rapidly divided tissue in your body. And in order to get a high enough level of chemo to penetrate into a tumor, you have to really load the patient with this, and that causes very broad systemic toxicity, which, as we all know, leads to significant collateral damage to normal tissue that undergo rapid cell division. So you lose your hair, your GI tract gets irritated, bone marrow, reproductive tissue. And then lastly, a common problem is that most cancers will develop resistance to chemotherapy after prolonged exposure. And this can be driven by a variety of things, but one of the more interesting mechanisms by which tumors evade or develop resistance is this induction of these things called efflux drugs transporters. And what will happen is that when the cancer is exposed to the drug, this protein efflux transporter gets expressed in the membrane, and it basically pumps out the chemotherapy drug from within the cell. So there's a low enough dose so it doesn't kill the cell, but it allows the cell a chance to develop resistance to it. And so and the other interesting thing is that you can use one chemotherapy drug which will induce that transporter, and it'll develop cross resistance to a whole bunch of other chemotherapy agents. So now you're really limited in what you can do. Okay. So moving on to now what I'm sort of getting to where I'm going to really talk about, but there's been in the last 20 to 30 years, I'd say, really exciting emergence of new treatment platforms. And this has largely been driven by advances in molecular genetics, genetic analysis, cell and protein engineering, and have enabled these therapeutic strategies. A lot of these platforms involve things like immunotherapy, which is what I'm going to be talking about with the antibody drug conjugates. A growing area is immuno-oncology, and basically what they're getting a deeper understanding now is the interplay between components of the patient's immune system and the tumor. And tumors have mechanisms that they can sort of turn off or quiet your immune system, and those things have been identified. And so when you block that signal from the tumor that turns off your immune system, you're sort of reactivating your immune system to better fight the cancer. Now yet this is a very delicate dance that you have to deal with because you don't want to overactivate your immune system because then you're going to have autoimmune problems. So we're constantly sort of trying, but there's a lot of effort and energy being put into immuno-oncology that's happening at AbbVie and other companies. So in addition to that, you have stem cell therapy, gene therapy starting to show a lot of promise, different types of laser treatment and photodynamic therapy, nanotechnology, which many years ago sounded like something out of the Jetsons, but is actually starting to bear some fruit, and it's a way to really selectively deliver things with these little nanoparticles to tumors. And so that's another area, hypothermia. And I'm just giving you a few examples of some of the main ones. So now, what I'm going to spend the rest of my talk or what I'm going to talk about are antibody drug conjugates. And basically, what this involves is a strategy to selectively deliver cytotoxic agents to cancer cells with minimal collateral damage to normal tissue. Any questions at this point? Or is it making sense or is it too much? Okay. Because sometimes I get caught up in the verbiage. It's a lot of verbiage. That's the problem with biology. It's like there's so much. It's hard to distill at times, but I'll do my best. Anyway, so ADC, antibody drug conjugate, also known as empowered biologics. It's an emerging class of a highly potent pharmaceutical drug that really marries immunotherapy, which is the antibody part, and chemotherapy, which is the drug part. Is basically comprised of three components. The big one is the antibody, and that's if I have this graphic here. I'm sorry, I don't have a pointer, but if you look at this Y-shaped thing, this is an antibody, and there's two long protein subunits, and then the smaller ones. And it's basically Y-shaped. This part of the antibody, I'm sorry if I can't see it, is what recognizes a tumor protein or any protein, and this is the backbone of the antibody. Okay. So again, so you want the antibodies designed to specifically bind protein that is hopefully only expressed or shown on the membrane of a tumor cell. Okay? Then there's the linker, and that is represented by this little green thing that's connecting this. This is the drug, this is the linker. Now that's actually a really important thing that has evolved a lot. That's a chemical structure that enables stable binding of a cytotoxic or chemo drug, which is represented by the red ball there. It's stably bound and it binds to the backbone or certain residues on an antibody in a way that disrupt the antibody's ability to do what it has to do. Okay, and then lastly, you have the drug, which is also known as a payload, excuse me, or a warhead, and that's a cytotoxic agent that is then selectively delivered and hopefully kills the tumor cell. So that is just a very rudimentary representation of what an antibody drug conjugate looks like. Okay, so now I I pulled this out of a paper. This is fairly rudimentary, but I thought I just want to demonstrate the mechanism of action of an ADC. And so the big circle represents, in a very kind of rudimentary way, what a tumor cell is, the plasma membrane, the outer membrane of a tumor cell. And so if you look, I wish I had a pointer. I didn't bring it. But if you look, this is our ADC right here. This is our target protein that is hopefully specific to the tumor cell. The ADC comes, this part of the antibody recognizes this particular protein. It is then goes through something called endocytosis. So when proteins are bound by antibodies, they form these vesicles, they internalize, okay? This then starts to migrate further into the cell, and it eventually gets down to a point called into an organelle called the lysosome, which is where the acidity is very high, and basically the antibody and all the components are broken down, which is typically where you want to have your payload released. So once that is released, now you have your drug either free or even bound by the linker, doesn't matter. It is now free to then bind to its target, DNA or whatever, and causes cell death. So that's just a quick explanation of how that works and why that's better than chemotherapy. Because chemotherapy can't specifically get into a tumor cell. This antibody allows that to happen. Okay, so it's kind of cool. I think it's cool. Um, yeah. So now I thought I'd go through what are the criteria that you really need to meet for an ideal ADC clinical candidate. So there's four components to this, and again, there's a lot I could go into here, but I'm just going to go over the main points. Tumor target selection is your first thing. You have to pick a protein that is amenable to making an antibody against, and typically you want something that's highly expressed. Expression means there's high levels of it. So like every protein in any cell of your body, there's a gene that makes that protein. And so when the gene is active, it means it's expressing a lot, it's making a lot of your protein. So you typically want a tumor protein that is highly expressed and that has either minimal or no expression, ideally, in normal tissue. It needs to be displayed on the surface membrane of the tumor cell, and which would enable the binding of the circulating antibody ADC. And then you want this protein to have something that when it is bound by an antibody, it has good internalization properties to enable the delivery of the cytotoxin inside the cell to the target to kill the cell and prevent cell division. The second criterion is your antibody selection. You need something that shows really good tumor target specificity and binds the protein you're interested in and not other proteins in normal tissue or off target. You want it to have high binding affinity. You want the antibody at a low dose to bind tightly to your tumor protein. And then you need that antibody to have good retention, meaning that it stays in circulation well, it doesn't get broken down too easily. It's also really important that the antibody you choose has low immunogenicity. If you inject an antibody into a human and that antibody elicits an immune response, the patient starts making antibodies against the antibody, that's going to neutralize it and prevent it from getting to where it needs to go. And so other things you need to sort of think about is you want that antibody to have low cross-reactivity, meaning you don't want it to bind to some other proteins in other tissue. Again, anything, properties that'll prevent it from having off-target effects. You want it to focus in on your tumor. And then it needs to have good linker binding properties. And I'll try and get through this a little quicker. Then there's the linkers. Linkers are, and there's medicinal chemists. This is where the people who are chemists at AbbVie do a lot of stuff with this. You have basically two types of linkers. There are the non-cleavable versus cleavable linkers, and that is largely governed by where you want your drug to get to. So you would want to use a non-cleavable linker to really prevent a really super cytotoxic agent from being released accidentally. So a strong linker with a dangerous payload is good, so it'll stay and it won't be released too soon. It'll get in way deep in the cell, go to the lysosome where it's finally released. And then you have cleavable linkers where that might be more useful if you want your drug to be released quicker when it's internalized and it can get to the site of action. Um, Broadly speaking, both non-cleavable and cleavable, they need to maintain the stability of the ADC in circulation. That's an important feature. It needs to, like I just said earlier, prevent the premature release of the cytotoxin. And then it also needs to enable, at the right time and in the right context, the release of the cytotoxic payload upon internalization or degradation in the lysosome. And then the last criteria is cytotoxic payloads or warheads. So obviously you want something that has really potent activity against a tumor cell that's able to kill it at a low dose. You want something that has fairly high stability in circulation or with more specifically inside the cell. If it breaks down quickly, it won't get to its target that it's supposed to hit. So the physical properties of the site of the payload, you want good solubility because there's an aqueous environment of a cell. So if you have a molecule that doesn't solubilize in an aqueous environment, it's just not going to interact with the tumor, excuse me, the target that you're trying to go for. You also want your payload not to elicit an immune response because that would affect its activity. And then you want low molecular weight because if you get these big fancy molecules attached to an antibody, that your body can have trouble dealing with that, and that can create issues for getting what where it needs to get. So strong payload with strong activity, low molecular weight, good stability, and then something called a long half-life. And half-life is basically the time it takes for something to be reduced by 50%. And so a long half-life means that your molecule is staying in solution for a long time. You don't want something that goes down after a couple days and becomes half of what it was originally, the concentration. That's no good. And so moving on, the payloads that are used currently primarily belong to two classes. One is microtubule disrupting agents, and the other are DNA damaging agents. And I just pulled this graphic in here. This is like an eighth grade, maybe high school, maybe college biology class. If you remember, you remember about it. So microtubules, cell division. You have before the cell divides, you have these things, these spindle fibers. And these are all microtubules. And they're subunits that polymerize and form these long spindle fibers. The chromosomes can line up here, and then they can migrate after they replicate and and then form the daughter cells. So microtubule disrupting agents are things that will bind to the subunits of these things and prevent them from forming the fibers. So you stop cell division that way. And then the DNA damage agents I sort of described earlier. Um, so I thought it would be good actually because it's a new therapy, but it actually has a kind of a bit of a history behind this novel approach. Actually, 100 years ago, there was a German physician scientist named Paul Ehrlich who first conceptualized what an ADC was and coined the idea of an immunotoxin. He called it a magic bullet. And he thought, wouldn't it be great if we could attach something to an antibody to get it delivered, not just tumor, but to any tissue that you're trying to sort of treat? 45 years after that, the first effort at making an actual ADC, they attached methotrexate, which is a chemotherapy drug, to an antibody that targeted a protein in leukemia. This was the first time an ADC was ever made. It didn't get very far, but at least it was done. It was the first effort. Moving on to the 1980s, people were actually now doing clinical trials of ADCs using antibodies that were generated in mice. And there were some clinical benefits shown, but mostly these things fail pretty miserably for a number of reasons. First and foremost is that the antibodies that were generated in mice, because they're mouse antibodies, when you inject that into a human, what's the human immune system going to do? It's a foreign protein. It elicits antibody against the mouse antibody. So totally neutralizing the activity of these things. Also, there was problems with weak stability in the linker chemistry. They didn't, at that time, there wasn't really that many options, and so those things would fall apart easily and release the payload easily. And then the third thing is, that, like I just said, the premature release of the toxic molecule, then causing unmanageable systemic toxicity. So the big change came in the 1990s, and this is when a lot of new genetic techniques and antibody engineering were coming into vogue, and a lot of possibilities were being opened up. And this is what is called the generation of chimeric or humanized antibodies, which enabled you to circumvent the immune problems caused by earlier versions of the ADCs where they were using the mouse antibodies. Okay, so what happens is, and let me see, I might have this in there. Okay, so here's a graphic to sort of demonstrate that. So in order to make an antibody against a human tumor protein, what they would do is you get that protein from the tumor, there's a variety of ways of getting it, you purify it, and then you inject it into a different animal, a mouse, a rat, maybe a rabbit. Mice tend to be what were mostly used. And that mouse, because it's a human protein from a tumor, will make a ton of antibodies against it. And so that's great, but you can't then take that mouse antibody and use that therapeutically in a human because it's a mouse antibody. So what they did with genetic engineering techniques is they would get your initial mouse antibody, which is in yellow. Murine means mouse. I'm sorry, it's a technical term. And with genetic engineering techniques, they can swap out these different domains here, and the human antibodies in red, and put those into what used to be the mouse antibody and do it up there. And now you have the part of the mouse antibody originally, and that still binds the protein, right? It's got the binding properties. However, there's enough stuff here that could cause an immune response. So they took it one step further, really turned this into a human antibody. And now you got just the ends, that's what binds the protein. That's the only mouse part. So it does not elicit an immune response when you put that into a human patient. Okay? Does that make sense? Yeah. Okay. All right. So now, and I know it's a lot to look at here, but I thought I'd go through what are the stages of development of an ADC. It's a multi-stage process, as you can imagine. The first step is you have to identify a suitable tumor target protein. And so that is what you do. And people just have discussions. You do some basic experiments, find out if this is something that you can make an antibody against. You then harvest as much of that tumor protein. I won't go into the ways, but there's things you can do. Like once you know the gene for a protein, for your tumor target protein, you can take that gene and clone it into a cell that is specifically used to generate tons of that protein. And it's like a factory. And then you take that protein and you purify it, isolate it. Now you got a lot of this tumor target protein. And then that takes you to step two, which is you inoculate or you inject it into the mice, and they generate tons of antibodies against that. And the first thing you do is you characterize the binding properties of those mouse antibodies, and you look at that in a panel of tumor lines that express your target of interest. And once you find sort of a group in step three of the high affinity binding mouse antibodies, you then initiate the humanization of those antibodies by what I just showed you previously, changing the backbone and making it more like a human. And once you've now humanized your mouse antibodies to the extent you need to, you then go back in, you reassess binding, and then you can conjugate those antibodies to different payloads and start to look at their ability to kill tumor cells in a petri dish. And that's largely what myself and several others do, is that we're step two, three. Um, and that's a lot of work. It actually, I mean, it's two steps here, but it's the kind of thing that, it can take time. There's problems. There's sometimes cells get infected. Sometimes they quit on you. It's not always so straightforward. Biology is a science of exceptions, not rules. It's like I like to say, because things are always happening you don't expect. And anyway, so a lot of work goes into that. Once you get that information and you've got a lot of good stuff there, then you can take those best ADCs now because they've got drugs attached to them, and you go into animal studies where the ADCs are tested in different rodent models of human cancer, and you look at their ability to shrink or arrest or stabilize tumor growth in the animals. Step five is just additional stuff that has to be done, looking at like stability of the ADC in the serum, looking at a toxicological effects in different tissues of an animal, and they have people who specialize in that. And once you get all that data together, the in vitro data, like I talked about, the binding, the cell killing, the in vivo data, looking at the effects in animal models, and then other properties, the pharmacokinetic properties, the stability, et cetera. Then you can then narrow down on a single best candidate from that, and you submit that to the FDA for what's called an IND or investigational new drug. And then now moving on to the last step, 7, if the FDA grants you IND status, then you can initiate clinical trials in humans. There are 3 phases of clinical trials, phase 1, 2, and 3. And you need to pass pretty high bars of drug safety and efficacy to eventually get through all of those. And this is typically where drugs go to die, because we can cure cancer in rodents all day, but you put it into humans and stuff crops up, side effects happen, it doesn't work like you expect, and you have to like let that go and go back to the drawing board. So step 7 is really like we're all kind of holding our hands and hoping it's going to work out. And sometimes things can look great in phase 1. They can look pretty good in phase 2, and then phase 3 something happens. There's some off-target effects that weren't apparent in the first 2 phases, and you're like, oh my God, what are we going to do with this now? And you have to park it and back to the drawing board. So I thought it'd be a good time. The average time it takes to go from step 1 to 7 is roughly 15 to 20 years, and that's if it all goes smoothly. So it's a long time and a lot of effort. Typically, 1 out of 50 candidates makes it from early discovery, step 1, step 2, to the IND filing in step 6. And then from that, 1 out of 5,000 actually gets from that beginning step 1 out to the market, 1 to 5,000. And so this is just a rough estimation, but the combined cost of the early R&D, development, clinical trials, production, and marketing is average 1 to 2.6 billion. Liz, you might be,
Yeah, I'm
is
glad
that about
that
right?
because
Yeah,
I think a lot of people don't understand how truly long it
it,
takes
it's long,
for a
it,
drug to
it
even
takes,
make it.
right,
So the odds are
right,
very slim.
yeah.
I just was gonna ask you maybe if you could describe what goes on in phase 1, 2, and 3, because I think a lot of people.
Yeah, so I should have spent a little more time. So phase 1 is basically it's a smaller group and it's looking at safety and efficacy and figure out what the maximum tolerated dose is. And it's a small group. Phase 2 is where you expanded it and you're looking at maybe a broader population of patients that would benefit from the treatment, and you're trying to build up some statistical significance in the effect, and trying to see, get an early read on some adverse events or side effects that might happen, and also demonstrate clinical efficacy. And then phase 3 is just a further continuation of that, basically. And then it's been in studies longer. Now you can sort of see, are the side effects something that could be mitigated, managed, and if they're not, then you have to park it, like I said. So I don't know. There's probably more to it than
Yeah,
that.
well, I mean, that was.
But
That's
so it's safety and efficacy. Is this safe? Can you put this? What happens when you put it in a human? Is this gonna cause something that we didn't expect? And that happens. Is it effective? And I've seen some phase 1 clinical trial data, and I'll get to that in a minute. And unfortunately, I couldn't bring it with me, but there was a brain scan of a 43-year-old guy that in a phase 1 clinical trial, this ADC that I'm going to get to in a minute, for glioblastoma, and he had a very sizable lesion in his brain. And after about 10 months on a fairly low dose of this ADC, the lesion was 82% reduced. And then in another like 4 or 5 months, it was not evident in the MRI. And that was like a eureka moment for us. We were like, holy cow, this can work. Unfortunately, went on in phase 2 trials. It didn't show as robust effect in other patients. And there wasn't a significant enough benefit. And so as exciting as that first data was, it died after phase 2. And I'm going to get to that, but it happens. And that's typically what you sort of feel like that Greek myth Sisyphus. You're rolling rocks up a hill just to have them roll down sometimes. And, you have to be willing to do that. Yeah.
There are certain cancers that are more susceptible to this technique
Yeah, you know what? Most cancers are. I think they're showing a very effective application in hematological cancers for one specific reason. Blood cancers are easy, there's easy access. So you inject your antibody drug conjugate into the blood, it's easy to get to a leukemia cell or myeloma or a lymphoma cell. Some tumors are more accessible, some really aren't. Like pancreatic cancer is a really terrible cancer for a lot of reasons. Everyone knows that. One of the things that pancreatic cancers do, which is really nefarious, it's just amazing. You start to wonder what's driving these things, but they will co-opt normal tissue to form barriers around the tumors. So if you got a like a moat of normal tissue surrounding, you can't get your antibody drug conjugate to where the tumor is. You have to break down that barrier somehow. People are working on that, but it's hard. It's tough. So it's systemic accessibility of your ADC. What are the tumors like? Is it well vascularized? Can you get your antibody drug conjugate there? So, yes or no? Brain cancer is hard for a couple of reasons. Not only do you have to get it to the tumor, but you have to have it cross the blood-brain barrier. And that is a thing that is selectively prevents things. You don't want a lot of stuff to go into your brain. And so you have to get this big molecule across the blood-brain barrier. Now, typically people with GBM have compromised blood-brain barriers. So that's one thing you can take advantage of. So it'll get there and do what it has to do. So that's a good question. Yeah. Okay. Any other questions? Yeah,
Something like CRISPR, will those, some of the new developments
sure.
enable
Yep.
parked drugs to suddenly maybe be, check, like will.
Sure. Well, CRISPR allows you to, and I'm not going to go, that's a very, everyone hears about CRISPR, and that is the gene editing technology that is latest and greatest. And it's a very useful tool for trying to understand what different things are doing to maybe knock genes out or knock genes in and see how you can maybe handle some of those hurdles for sure. Yeah. So, all those genetic editing techniques can be used in a variety of ways to try and deal with some of these things. Okay. Any other questions on this or?
Like the advance, I see like 15, 20 years, and we got bigger computers and things like that. Have advancements over time made that time shorter, or do you see that?
I think, yes, this is just based on stuff that's now on the market now, but I think with the use of bioinformatics and strong computer systems to do bioanalytics and really look at things, that'll save time and money for sure. That's going to be a big thing. AI, can be utilized to sort of help sort of shorten the timeline on things. But at this point, this is just what's been in the last 20 years, things that start, so something I worked on back in 2008 got to clinical trials in 2018, 2018, so that's just kind of the normal way it happens. Okay, so I'm going to move on. Now, as great as these ADCs sound with great clinical potential, there are significant hurdles that remain. Um, like chemotherapy, cancer cells can develop resistance to ADCs after long exposure. And this resistance can be mediated by a whole bunch of mechanisms involving all components of the ADC. Among these, you can have the tumor may downregulate or reduce the level of the tumor target protein, so the antibody no longer has something to bind to. That's one thing that happens. There can be changes and alterations in the trafficking pathways that affect the way the ADC is internalized and processed within the cell. There can be more general changes in the cell cycle machinery and regulatory signaling pathways that can affect the way the ADC does its work. And then there's also what I mentioned earlier with chemotherapy, the removal of the toxic payload by these efflux pumps that pumps it out of the cell so it can't kill the cell. And then you can even have further modifications or things within the cell that will enzymatically change the payload and render it inactive. So the lack of potential or the inhibition of the activity of the ADC can be a combination of all these things or each one individually. It's very complicated. And we try to look at and understand that. In addition to that, you can have ADCs causing other things and side effects that can underestimate or undermine their therapeutic potential. And this comes to light in clinical trials. Things like neutropenia, which is destruction of your neutrophils, thrombocytopenia, which is the destruction of platelets, skin problems, ocular tox, neuropathy. This is just a few. And ocular tox is something that happened with what I'm about to talk about that actually really unhinged its development. Severe problems in the eye. Um, However, these liabilities that I just mentioned are being addressed with new optimization strategies, and those will hopefully come to bear on ADC development and to sort of get over these hurdles I just mentioned. So they're constantly working on that, finding new strategies to sort of overcome these things that can happen. Okay, so that's the background of ADCs. Now I thought I'd go to a specific thing I actually worked on starting in 2008, I think 2008, called ABT 414. I've mentioned this. This is now public domain, so I can talk freely about it, but I've spoken about to people here at different functions. They ask me, what are you doing at AbbVie or Abbott? And I'm like, well, I'm working on this thing for glioblastoma called ABT 414. It's an antibody drug conjugate. So that what this was developed for is to treat and target glioblastoma. GBM or glioblastoma is a fast-growing malignant brain tumor. It affects the brain and spinal cord. It's a very common and aggressive tumor in adults. It's got a terrible prognosis. Average life expectancy is actually 12 to 15 months. There's roughly 12,000 cases diagnosed in the US each year. The symptoms, and this may be not all of them, but the main ones are headache, nausea, drowsiness, blurred vision, personality changes, seizures. So it's a brutal thing. And the standards of care for GBM are not great. Surgery is really difficult when you're dealing with brain tumors because it's snakes around normal tissue. So it's not easy stuff to remove. Some tumors are easy to get out. They're like just a ball of a mass of a tumor. Like brain tumors like this sort of has like tendrils or things, and it's really hard to remove by surgery. Radiation's one thing that's used, and that's, to some success. And then the only chemotherapy that is used for GBM is this stuff called temozolomide. And I've worked with it on GBM cells. It's worthless. It's just not a good chemotherapy drug. It doesn't do much. And then there are just palliative treatments to help ease the the symptom of burdens that people suffer from. Interestingly, GBM or glioblastoma cells do express a very unusual form of this mutant growth factor receptor called EGFR, epidermal growth factor receptor. And EGFR actually is a growth factor that drives a lot of different cancers, and GBM is one of them. But in GBM, it has this weird mutated form. It's called V3. There's basically a deletion of some components of the growth factor. And it is constituent. What that results in happening is it really drives a tumor to grow out of control, and it's always active, this growth factor. So that makes it, because it's so unique, it's a unique signature of these cells, it makes it a really attractive ADC target because it's highly expressed in GBM and basically not expressed in any normal tissue. So ABT 414 was this ADC we developed, and it selectively targets that EGFR V3 in GBM, and it was used to deliver this payload MMAF, which is a microtubule inhibitor, if you remember what I talked about earlier. So now I'm actually going to show some real data. This is from a paper that is in the public domain, it's stuff that we worked on, and it showed this is in vitro activity. So on the left side there, that is showing the binding properties of, it's called ABT 806. That's the naked antibody part of ABT 414. And this is looking at its binding to a GBM line called LN18 that expresses the EGFR V3. And as you can see, now there's included here is a positive control antibody called cetuximab, and then a negative control antibody called rituxan. And you can see in blue here, really nice high binding of 806 to the V3. So that's good. As well as high binding, then we also demonstrate now it's ABT 414 on the far right. That is the 806 antibody with the microtubule inhibitor attached to it. And you can see in another GBM line that it really is effective at killing the cell at a pretty low dose. This is between 1 and 10 nanomolar. I don't expect anyone to know what nanomolar means, but that's just a concentration. That's a low dose. So you see effective killing almost, way down. This is a control antibody. It doesn't kill with the same payload on it, MMAF. This is the antibody with MMAF, and it really is doing a nice job killing. So that's the kind of data we would generate, and we would do that in a whole panel of different GBM tumor lines. this is what it looks like in vivo. Now, this is the kind of data that kind of really, like what I do is exciting and important in a way, but what really is exciting is when you put this stuff into a rodent model that have been implanted with human tumors, and you see that the tumors get flatlined. That's kind of gives you some hope that we have something that's meaningful. So these are rodent studies. These are mice that have been implanted with human tumors. So there's A431 and A, that's a human tumor. H1703, another GBM line. HCC827, and then SCC, that stands for squamous, it's a head and neck cancer cell line. They all have high levels of the EGFR protein I was telling you about. And as you can see, at a fairly good dose of ABT414, which is in the dark red circles, in all cases, you get this incredible flatlining of the tumor. It's not growing. This is the vehicle, these are the control antibodies, these are lower doses. That tumor is going quickly here. ABT414, it's not growing. That's great. Same thing in this line. You can see, really stand out here. The lower dose at 3, also doing a nice job. It's starting to grow back a little bit. At 10, it's not. Down here, same thing. Here, not as significant, but still outperforming the other antibodies. ABT414 looks great. So that's the in vivo data. You put that together, and we're all excited. Strong in vitro and vivo data package enabled IND filing. I guess it was 2012. I might be wrong on that. It might be 2014. I think 2012. What I was hoping to show you today, and I'm really bummed because this would have been my ooh-ah slide, was the phase 1 clinical trial data, where it showed unbelievable reduction of tumors in several patients. And I couldn't find these scans. I asked people. I don't know if it's under lock and key. It's not in the public domain, so you just get to take my word for it. It was really dramatic reduction of this tumor in this 43-year-old guy. It was like unbelievable. So that was exciting. Because of that, those strong results in the phase 1 trial, that accelerated its entry into a phase 2 clinical trial. This is a larger cohort of patients, some having the V3 EGFR V3 amplification, that protein I mentioned earlier, and there are some people with GBM who don't have that, so they want to have a combination and see what happens. And as it turns out, and it's often the case, the results from this phase 2 trial did not recapitulate as what we saw in the phase 1. It did not show overall survival, which is OS stands for, in most of the patients. So the decision was made to discontinue development of ABT414 or shelve it for the time. Any thoughts or questions yet?
So
Very good.
with the technological advancement that has happened, do these things actually come back? They say, oh,
They might, yes.
they used
So
to
that's why we
work
shelve them. We don't say,
with
yep,
say,
yep.
oh, we can figure out why this.
Yeah, yeah. So, and interestingly enough, like I've been off of this project for 10, 12 years now, because I've moved on to other things. But I have doctors and collaborators from Mayo Clinic, from Germany, from um, Sloan Kettering, who we sent material 414, and they were using it in studies with patients, and there are patients who responded well. They're really upset that we're not working on this anymore, because for certain people it worked great. And they still ask me we still have in a -80 in my lab, I've got vials of EDT414. And I'm often asked by my old boss, like, hey, can you send 50 milligrams of that to so and so at whatever so they can continue these studies? So there's still like this percolating interest. I mean, to your point, it might come back, once you can sort of figure out something. Yeah.
Is this something that's offered to patients that have leukemia, or is it just?
For GBM, no, you can't offer it. Unfortunately, no, I don't think so. No.
It's not actively being offered.
No. No, unfortunately, but it's being used in studies. If you're included in a study with a group at Mayo or whatever, they can give it to you. So that was disheartening. Oh, go ahead.
The massive amount of money to kind of get through these first few phases, and then you kind of put it on the shelf to see what happens. Fast forward 10 years, and people from other parts of the world are like, hey, can you send me some of that? I'm trying to,
Yeah,
and technology is constantly developing in all sorts of aspects
Right.
here. How does that work, and why would the institution that put in all this money, other than I want to make the world a better place, talk to me a little bit about the willingness to share.
Yeah, well, that's a very complicated thing. And I'm not surprised. That is it's a dicey thing. You have to so we have agreements, we have people at AbbVie that, and you have sort of things that will sort of prevent you from just giving stuff away, and so is that kind of what you mean? Like,
Yeah,
yeah,
what's you get this, I want to make the world a better place, I want to advance this technology, and then you fight that against the massive amount of money that's put in by private to create technology that may not have gone to the finish line, but it went somewhere.
yeah,
And it just seems like so many of these little pieces are probably all floating around there with
they are.
a low desire to link the knowledge
Yeah.
to help the world.
Yeah, that's a good point. There are other people that are more involved in the strategic alliances and things like that that could answer that much better than I can. But that is definitely,
You're in the lab.
I'm in the lab. I should know, but it's always good to know about
It's
it.
an interesting.
It is. It's very,
I remember when our daughters speak talked about you're learning all these technologies, you're creating all this great information,
yeah,
have some responsibility. Where does it go? How does it apply?
yeah, yeah,
And it's there's a challenge in how it all.
for sure. Yep. Yeah.
Their collaboration, agreements with institutions and
Yeah.
AbbVie that settle things like intellectual property,
Right.
address those
Yeah.
sort of issues. So
So-
they do come to an agreement,
Right.
which are mutually beneficial.
And they have to constantly be, so like when I'm sending material overseas, like we had a collaboration with a group in Israel, and the importation documentation and all the sort of components of this agreement were profoundly complicated. I'm reading through it all and making sure I'm checking this box, checking sure the collaboration is up to date. And so and we have people in legal that we send that stuff to, and they have to sign off on it. And so it's a pretty formal, they're pretty on top of this. So yeah.
When you have a drug like you had your phase one, that you got excited, and when you move to phase two, it's not as exciting.
Yeah, I'm gonna move on because this is the phase 2 data.
Okay.
But anyway, go ahead.
Do you, now with the advent of the new computing models in bioanalytics, is there a way to, if you will, analyze the failure to look at where the strengths and weaknesses?
Oh, for sure. Yeah.
So that you can
hmm
design to compensate or design.
Yeah,
to
but more evolved clinical trial design for sure. Yeah. So that sometimes clinical failures are because you just didn't design the clinical trial very well. You didn't stratify the patient population. But you could use like computer models that help come up with a much more enlightened, effective clinical trial design that might get you to a better place and not having you scratch your head like what's going on here. So yes, for sure. Computers will help with that. Mhm. So anyway, this is the actual data from that phase 2 clinical trial. I know it's kind of hard to look at, 66 patients. And what this is just time on therapy and then different sort of responses. So out of 66 patients, there was one guy who, and then a little plus sign here says that they are positive for the marker. We're interested in the EGFRV3. And then you have X means that they went off the trial because they were having adverse events, some problems. So you see a lot of crosses, people getting off. You see a mixture of pluses in different parts here. This complete responder had the D3, so that's kind of what we saw in phase 1. Then you have three patients with partial response. And then a nice significant number of guys with stable disease, and then all these up here had progressive disease. So this is not terribly great, And on top of that, this is a table with all the different adverse events or side effects they had. The biggest one that really unhinged this, so there's non-ocular issues, fatigue, headache, diarrhea, some other things. But the real issue is this ocular toxicity. Now your eyes have a lot of EGFR receptor in them. They're not sure if that's what it was, or there's just something about the payload itself that somehow got into the eye and caused terrible problems. If I showed you pictures of these eyes, it look like something you can't believe. Just cloudy, like it's just the worst thing you've ever seen. And so, blurred vision, dry eye, keratitis, photophobia, eye pain. A lot of these people had it. So that, together with a lack of response, you can't go forward with this,
Yeah.
right?
But at the same time, the brain tumor could be in a place that would be generating those problems,
Yeah,
not the solution. So like wherever those tentacles went in the brain could be causing the problem of the ocular toxicity.
that's a possibility. That's a possibility. Yeah. The only thing is that regardless of how extensive the brain tumor was, it wasn't a perfect correlation. Like if you saw ocular tox with people with really broadly disseminated brain tumor, then you could make that sort of connection. But it was happening in people with low levels of tumors, and so it wasn't a perfect correlation. It was really all people had some level of ocular tox, regardless of the status of disease or tumor burden. So, yeah,
Which she could the
yeah,
develop with delivery of the
sure. Yeah, I mean, yeah,
and maybe turn this.
that could, yeah, for sure. And people are working on that. Yep, yep. I'm not, that's not my background really, but I know that that is a growing area and growing possibilities and modality to treat. And so anyway, that was a gut punch, like I thought we were gonna high five and have beers at Mickey Finn's, but phase two said, no, we're not gonna do that. So anyway, in spite of so ABT 414 is an example of often what happens. Nonetheless, there are ADCs that actually do get to market. So here I've just thrown up this table here, and this shows you some of the ADCs that are currently being used. It's a lot of big players in the field. If you look in the second category, you got Pfizer, Genentech, GSK, AstraZeneca, Daiichi. So everyone's in the game, and they have things that are now, in the market, and a lot of them are blood cancers, AML, B cell, DLBCL, so another multiple myeloma, large cell B cell, and then HER2 positive, that's a breast cancer, and some other triple negative breast cancer, et cetera. So it's happening. There's things that are out there. Right now there are, I think, 15, maybe a little more currently in the market being used, and they're, like I said, targeting both blood and solid tumors. And then there's actually in clinical trials, and I don't have this point here, there are currently over 100 ADCs in various phases of clinical trials by a wide range of companies. So it's ADCs are happening. They're going to be a tool we can all use and that clinical oncologists can use to treat their patients. So that's kind of it. So to summarize, the antibody drug conjugates provide a new strategy to fight cancer. They target specific tumor proteins, enabling selective delivery of the cytotoxic agent to cancer cells, sparing normal tissue. Continuing advances in antibody engineering, linker chemistry, better cytotoxic agents are yielding new generations of ADCs with better therapeutic potential. And like I just said, I just showed you there are currently 15 ADCs in the market, over 100 in clinical trials. So I will say that my last statement is that these things continue and will be a continually important tool or arrow in the quill of clinical oncologists to better serve their patients. And that's all I have.
Next comes at
So,
1:30, but we can take a couple of questions.
yeah,
I was just wondering, can the nature of the tumors change, and do you look for changes in the tumors themselves?
absolutely, for sure. They do change. So you can look, and I remember sharing this with my dad. You can look at the genetic profile of a primary tumor, and then you can look at the genetic profile of metastatic lesions that came from that primary tumor, and they might share 20% of the same genetic profile of activity. So that's another added burden. Biological systems are profoundly dynamic. That's the problem. It's like things are not hardwired. So you can knock out one pathway and some completely unrelated pathway will then take over and compensate for that. And it's something you would never expect. And so we're trying to keep on top of all that and manage that kind of stuff. So yes. And so what people are doing is, and really, I think more effort should be put into understanding the genetic transitions that happen that make cells in a primary tumor leave the mothership and form new tumors. And there are people who are working specifically on that. It's very complicated, but that to me is a therapeutic possibility that would maybe be the most effective, you know.
Yeah, the standard procedure with a patient or not.
I don't know the answer to that. Not sure.
There's one more question.
Yeah.
Getting back to the mechanism of how that payload gets into the cancer cell, once it's done its job that you want it to do,
Yeah.
cell ruptures, dies.
Yeah.
So that payload is just floating in the intercellular
Yeah,
space.
it depends. Typically not. Once when it's bound, because these things bind with really tightly to whatever that is, and it'll stay bound. And when it's bound, it's not active, and so it's just broken down, metabolized, and either eaten up by part of your immune cells that get rid of that kind of stuff, or so. But it can some payloads can float around and cause have an effect called bystander effect. So they might, if the drug is released from the ADC quickly and somehow it leaks back out of the cell, it can go then and bind to cells around the tumor, things in the tumor microenvironment, and that could be a benefit actually, or it could be a liability. So that's a very good question.
Well, thank you
Yeah.
for doing. I'm glad we
Well,
learned science.
I just want to yeah.
Thanks for coming, everybody.
Yeah. Great. Yeah. Thank you. That's great. Thanks for
I've got a present for you. Fantastic. There's no point. Perfect. Thank you, sure. Job. Thoughts. Like, I spend my opportunity and Steve. Hey, thank you. It's fast. Thanks for Hey, how are you? It's okay. I'm glad you shoot him. Oh, what do you think? I know I was trying to get it on campus. And I we did it late and my computer again here. Right, so you're doing it. So you're good. Thank you. Stop going my fly. Mr. Freezing these guys. Good boarding school. Yeah. Yeah. I'm sure someone come back. Okay. I gotta say, let me get up smart people that are worth like that.
How about Russell and Spencer?
It's got good size teachers.
So what?
How is
They're all on the youth, right?
what? St. George.
Oh, okay. Russell's on the youth.
Right.
Okay.
Is that for Taylor? He's at St. George, yeah.
Okay. And you got Porter here. Yeah, a lot.
I like trees.
Oh.
Send that to Tucker Carlson.
Just want to say thanks for a great presentation. So,
Kevin,
Oh, no.
Yeah, buddy, thank you, man.
I've never heard so many questions during presentation. So that was did a great job. So, excellent.
Pleasant to see.
It's interesting stuff. And, that's why I kind of like the science things like you can't get cutting edge stuff. And sometimes when you look at it, it's going to help.
Yeah, so Joel's husband
Oh, okay.
and he does a lot of similar research. Work with viruses.
Okay.
And
Oh, very cool. That's the first stated, yeah. Nice, yeah. I know. It's a little, it's like kind of like, wow, it's crazy what people are doing. The viral stuff is amazing. I should have touched on that a little more, but again, I'm like, I can't go into all that. It's like waiting.
Yeah.
Like I can't do it justice in 2 minutes.
And you're a great person, like you can stand who I think is very bright because
A little bit, I know. I'm so used to doing. Right? Yeah, it's yeah. Yeah. I get the same response when I'm hearing finance. I'm like,
yeah, why? Believe me, I sit there, I'm like, fine.
well, thanks for that.
All right.
The problem. Might be rough. I just want to heal them. Hold on. Say.